All right, in this lesson, we're going to talk about how enzymes are impacted by their environment. There are some guided notes for this. Hopefully you've downloaded them and are ready to take some notes. So, just as a review here, this is a diagram that comes up quite a bit. The gray molecule here, this is a macromolecule, this is the enzyme. Enzymes are always proteins, and they can break down various substrates. They can also form different substrates or different reactants. Substrates can be anything, right? They can be carbohydrates, they can be proteins, lipids, nucleic acids, and even things like plastics we're finding out that can be broken down. The active site is where the substrate binds, and the shape of this is incredibly critical because the substrate has to fit in directly, almost like a Lego, into the protein. If it can't fit in there, then it can't come in physical contact with the enzyme, and the enzyme cannot do its work. So if we're going to break down a substrate, think about like if this is a protein in your gut or a lipid, and it can't bind to the protein, you're going to have undigested proteins going through your body. That means you are not getting nutrition, and probably a lot of problems will happen there. Okay, so the shape of the active site is incredibly critical for enzymes. In order to understand how we could get a shape, we might need to go back to how proteins are formed. Okay, so proteins are formed in the ribosome, and that ribosome itself is a protein that makes up an organelle in our cells. Okay, so the proteins will receive instructions about how to make a particular protein from the DNA, which gives it to the RNA. The RNA is, remember, that's kind of like a post-it note copy of the instructions of the recipe, and the ribosome will put the blue amino acid next to the orange, next to the green and the yellow, and in a very prescribed order will connect all of these amino acids, almost like a chain in a necklace. Okay, so it starts off like that. And what we need to know here is that each amino acid has different chemical properties. Okay, some of them might have hydrogen bonds hanging off here. Some of them might have sulfur molecules hanging out, and it would kind of be like if the green ones had sulfur, they're going to stick to each other because sulfur likes to connect with sulfur. The same thing with the blues. Okay, hydrogen bonds can form between two hydrogen molecules. So what happens once this long strand of amino acids is formed is that those molecules start to interact, and they make these little loops and kinks in the strand. Okay, and that forms what we know as helices, or sometimes if they're really close together, a sheet. Okay, these are two-dimensional helices or sheets that form from that long strand, and those in turn become this three-dimensional structure through folding. We call this protein folding. Okay, so they create this big glob of a protein, but because of their interactions between the sheets and the helices, they fold in a particular way. Okay. When you have multiple subunits of these proteins coming together to make one protein, that's what we call quaternary structure. This is tertiary structure. Okay, so some proteins are made out of multiple protein subunits, but that's a little bit beyond what we need to know for this. Okay, but this is how proteins form. Now, this origami crane was made by folding this piece of paper, right? This piece of paper is modified in a way to make this crane to function in a certain way. It gives us the image of a crane, of a bird, and you could even maybe put a Lego guy right here to ride the back of the crane. If you were to unfold this, right, you could definitely do that. You could do it without ripping the paper apart, but that would change the function. That would change the shape of the bird, of the paper. And when this happens to proteins, when we take something like this and unfold it to its original structure, that is what we call denaturation. Okay, so protein denaturation essentially changes the shape of the active site or the protein itself by unfolding it. Okay, so this might be a little easier way to understand how the functionality of the protein changes through denaturation. If you were to put a Lego in a microwave and it melted like this, you would no longer be able to use that Lego to connect with something else, because the active site, right, the active site where it connects to another molecule, the substrate, has changed entirely, so it cannot connect anymore. It is pretty much useless. So what kind of things are able to denature a protein? Well, there are certain environmental factors. We're going to focus on two in this class, and the first one is temperature. Okay, we all have optimum temperatures that we like to that we function well in, right? I don't mind 50 degrees, but I would prefer 60 or 70. Or 75. Probably 75 is a really good temperature for me. I don't like it above 90 unless it's really dry. So apparently I have humidity optimums as well. Proteins have optimum temperatures where they have the most functionality. And when we go beyond that, when we heat them up too high or don't give them enough thermal energy, then we can denature them. Okay, so the egg has a lot of protein in it, and what happened when we cracked it into a hot frying pan was that we unfolded the protein. Okay, so it is no longer one of these guys. It's not functional anymore. We essentially killed all the interactions between these molecules, and so you have a bunch of denatured proteins in this frying pan. Okay. Another way that we can denature proteins is by changing the acidity of the environment. And a lot of so proteins have optimum temperatures, but they also have optimum pHs. And what we mean by pH is the acidity of the environment. Okay. Our stomach acid, gastric acid, is one of the most acidic things that we might ever encounter. Okay, luckily we don't come into contact with it, and the inside of our stomach lining is made to not dissolve, thankfully. Okay, something that's a little less acidic than that is lemon juice, if that gives you any reference point for how acidic your stomach is. If you were to have a paper cut, for example, and you drop some lemon juice into it, it would sting a lot, right? A little less with apple juice, a little less with tomato juice. And so we have this acidity kind of decreasing, okay? We get to pure water, and this is what we call neutral on the pH scale. You'll learn a little bit more about all of this in chemistry next year, but you just need to know that acidity, okay, and pH matters for enzymes functionality, okay? If you go beyond neutral, we call this basic or alkaline. Okay, a lot of these things, I think about like bleach or ammonia, chlorine. If you dissolve chlorine in the pool water, it's very alkaline or very basic. And we do that intentionally so that we can kill bacteria or their enzymes, right? Or human cells that we don't really want them to be doing really great in the pool water that we're trying to swim in. Okay, so as is the case for temperature, all enzymes have optimal ranges where they function well. Okay, so the optimal temperatures and pH vary for each enzyme. Okay, the stomach enzymes are going to have a different pH optimum than the enzymes in our mouth, right, in our saliva. And this depends on their function. What do they have to do as a function, and where would we find them in our bodies? Okay, it's the same thing for where we might find organisms in the world. Some are found in really hot climates, others are in really cold climates. We're going to see an example of some of those in just a second. Stomach enzymes prefer a very acidic environment. Amylase, which some of you experienced if you eat starch, right, rice or crackers or bread, your amylase enzymes in your saliva will break down the carbohydrates. These prefer a very neutral pH, okay? And let's say this is temperature, but the optimal temperatures vary by enzyme as well. And what we're going to see for these optimums are this is where they function the best, right? The rate of enzyme activity. Sometimes we can have indicators, we can see what's being produced, and we say, oh, this enzyme is very active, right? But just because this is the optima doesn't mean that it's not able to act a little bit beyond that, right? If this were 73 degrees Fahrenheit, okay, I would be maybe optimally active. Okay, that's maybe my favorite temperature, but I'm still pretty functional until it gets to about 100 degrees, right? But my functionality definitely goes down when it gets hot, and the same thing like leading up to it. I don't function well at super cold temperatures, but I start to increase up to my optimum. All right. If we see the presence of life somewhere, you need to understand that there are enzymes there. Remember, enzymes are the proteins that are directing traffic. They are allowing for cellular respiration and photosynthesis and transpiration and all of the different processes to occur in the bodies. Okay, if an organism is tolerant of a certain environment, it is that way because the enzymes are tolerant. Okay, it means the enzymes are still active. We have this green Arctic sea ice. There's probably some sea algae that has adapted, and its enzymes have adapted to really cold temperatures. Okay, they're still able to photosynthesize. That's pretty amazing. There are also bacteria and archaea and different organisms that live in the hot springs of Yellowstone. Okay, if I put my hand in there, it would be burnt very severely, but some bacteria actually really thrive in these environments. So it all comes down to what the enzymes prefer and how they are able to withstand extreme temperatures or not, extreme pH. Okay? You don't want anything too acidic, but some, like these bacteria, apparently are okay with very acidic, and maybe they would not be okay if we put them in straight up cold water. Okay. So yeah, the presence of life should indicate the presence of enzymes, and because they are doing all the work. Okay. Hopefully that made sense. Thanks for listening.